Namaste students, Hamesha Khosrow I am JVN Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. So today we are here to discuss the topic, that is the essence of activation changes in the organization of the egg cytoplasm. So this topic comes under the subject developmental biology. So let's start. So we know that the fertilization process is having very specificity, means specificity of complementary to the gametes. So if the gametes are species specific, then only the fertilization will occur. So the eggs coating the zona pellucida, it is having an important role for this specificity, because zona pellucida is different from that of the another species. A specific species have a specific zona pellucida layer, and that is a characteristic of a species. So it plays an important role in the binding process between sperm and egg. So example, among the Kindremer solution of the jelly coat clam or agglutinate only spermatozoa of their own species. So if the only their own species of the gynodermates will have the fertilization process. Binding between sperm and egg of different species may occur when the zona pellucida of the egg is removed. So it is possible to fertilize the gametes of the different species if the layer of the zona pellucida is removed, because the layer creates the specificity. If the layer is not present, the specificity will not be there. So the gametes of the different species may be fertilized if the zona pellucida layer is removed. So there is a term, that is the monospermy or the polyspermy. So most animal eggs are having the monospermy process or monospermy phenomenon. So what is that? That only one gamete will fuse with the counterpart or the other gamete. So the protection is due to some properties of the egg surface. It happens that only one sperm or one male gamete will fuse to one egg cell. Not more than one sperms will fertilize with the one egg. So why this protection? Why the polyspermy is inhibited? So many phenomena are included in this process. One of the feature which is preventing the polyspermy is the egg surface. So egg envelope are responsible for this purpose, so this phenomenon. So molecular rearrangement of the egg surface that occurs during the egg maturation, that is the oogenesis, is responsible for preventing the polyspermy. So although immature sea urchin eggs have the ability to associate with spermatozoa, they also allow multiple penetration. So they are unable to develop the polyspermy preventing reactions. So since the mature eggs of most animals are fertilized before completion of the meiosis and are able to develop a polyspermy preventing reaction species, specific properties of the egg surface must have differentiated by the time meiosis stops, which is when the egg is ready to be fertilized. So one property I have told, that the egg surface or the property of the zona pellucida. So that is when a spermatozoon has started to move through the zona, it does not allow to penetrate of additional spermatozoa or zona reactions. Means that when one sperm is fertilizing the egg, so few changes are there in the zona pellucida which prevent the additional additional fusion of the male gametes. So although the eggs of some kind of animals are naturally polyspermic, only one spermatozoal nucleus fuses with an egg nucleus to form a zygote nucleus. All of the other spermatozoa degrade. Spermatozoa means you can assume it a male gamete. Okay, so there are polyspermy is also present in many species, but what happens that only one male gamete remains alive in the egg cytoplasm, and rest of the male gametes dissolve in the egg cytoplasm. So one thing is the fertilization membrane. Okay, so the most spectacular changes that follow fertilization occur at the egg surface is the fertilization membrane. So it is an immediate response to fertilization is the raising of a membrane called the vitelline membrane from the egg surface. So in the beginning of the membrane, it is very thin. Soon, however, it thickens, develop a well-organized molecular structure, and is called the fertilization membrane. So what is that? That before the fertilization, this membrane is present, but at very thin layer, as a very thin layer. As the fertilization occur, the membrane rising and it swells, and it called a vitelline membrane. Okay, so it is very thick and well organized molecular structured membrane. So at the point of the outer surface of the sea urchin egg, at which a spermatozoa attaches, the thin vitelline membrane becomes detached. Okay, so the vitelline membrane detached from the uh, portion where the spermatozoa attaches with the sea urchin egg. So as a result, the membrane of the cortical granules come into contact with the inner aspect of the egg's plasma membrane and fuse it with it. The granules opens and their contents are extruded into the perivitelline space. That is the space between the egg surface and the raised vitelline membrane. Part of the contents of the granules merge with the vitelline membrane to form the fertilization membrane. So the plasma membrane surrounding a fertilized egg therefore is a mosaic structure containing the patches of the original plasma membrane of the unfertilized egg and areas derived from membrane of the cortical granules. So these events leading to the formation of the fertilization membrane are accompanied by a change of the electric charge across the plasma membrane, so referred to as the fertilization potential, and the concurrent outflow of the potassium ion. Both of these phenomena are similar to that of occurs in the simultaneous nerve fiber. Okay, so there is a outflow of the potassium ions and the inflow of the hydrogen ions inside the egg. So another effect of fertilization on the plasma membrane of the egg is a several fold increase in its permeability to the various molecules like the ions, potassium ions and the sodium ions. This change may be the result of the activation of some surface located membrane transport mechanisms. So next is the formation of the zygote nucleus. So how the nucleus of a zygote is prepared by the male gamete and the female gamete nucleus. So after the entry into the egg cytoplasm, the male gamete or the sperm nucleus, now called the male pronucleus, begins to swell. Okay, it swells by absorbing the water and other nutrients, and its chromosomal material disappears and becomes similar in the appearance to that of the female pronucleus. Okay, so now the female nucleus and the male nucleus is known as the female pronucleus and male pronucleus. So although the membranous envelope surrounding the male pronucleus rapidly disintegrates in the egg, a new envelope promptly forms around it. And the male pronucleus, which rotates 180 degrees and moves towards the egg nucleus, initially is accompanied by two structures, centrioles, that function in a cell division. So after the male and female pronucleus have come into contact, the spermatozoal centrioles give rise to the first cleavage spindle, which proceeds division to the fertilization egg. And in some cases, fusion of the two pronuclei may occur by a process of membrane fusion. And in this process, two adjoining membranes fuse at the point of contact to give rise to the continuous nuclear envelope that surrounds the zygote nucleus. Thank you, and hope you are satisfied with this detailed session. If you have any query, please write in the comment box. I will resolve your query. And this session is powered by Digital Version 2 Jyoti Vidyapeeth Women's University Jaipur. Thank you.